Hello and welcome to the review video for Genetics Chapter 4, which is all about biotechnology. So we can start with the introduction to biotechnology, and the first thing we need to think about is forward versus reverse genetics. So basically, forward genetics means going from the genotype to the phenotype, whereas reverse genetics is going from the phenotype to the genotype. So, how forward genetics works is that when we go to phenotype to genotype, we observe a phenotype, say like a disease, eye color, hair color, something observable, and we want to see what genes are responsible for this trait. And then we look backwards and we try to find what gene is responsible for this trait. Reverse genetics is when we have a gene and we don't know its function, and we try to find what is the phenotype associated with this gene. So, does this gene correspond to eye color? Does it correspond to hair color? What does it correspond to? So, those are the two different types of genetics, and we have a lot of different techniques for forward genetics, and we have different techniques for reverse genetics, and sometimes we have the same for both. But in general, this is terminology that you definitely should know. X-ray mutagenesis we use in forward genetics to screen for phenotype, and then we can go back and use that to find a gene associated with that phenotype. And site-directed mutagenesis is we change a gene and see how that affects the organism. So that would be reverse genetics because we're working with the gene first and then looking at the phenotype second. Now we're going to go over techniques. So first we have cloning. So cloning as its basis is just making a copy of something. And so we have molecular cloning, which is what we usually do. Most cloning is not cloning an entire animal like Dolly the sheep. It's usually just molecular cloning, which is copying just one gene of interest or abbreviated as a GOI. And we usually use that to take the gene outside of the organism and manipulate it either inside another organism or we can sequence it or what have you. So, in order to keep, if we want to analyze what the gene does, we can put it inside a host, or as we call it, a vector, and we can use that in reverse genetics to understand what the function of the gene is and how it contributes to the phenotype. So again, this is an example of reverse genetics. And we usually insert this gene into a bacteria via bacterial transformation. And then we also use PCR to clone the actual um, gene. So we make a ton of copies and then we put it into this bacteria. PCR is the heart of molecular cloning and what we use to make lots of copies of a snippet of DNA, and around one gene of interest. So we have primers, which are the binding sites for DNA polymerase, and they specify what exact segment we want to replicate of the larger DNA segment that we have. So we have two types of primers. We have the forward primer and the reverse primer. One thing about primers is that they always have to be in the 5' to 3' direction to guide the DNA polymerase to where to start synthesizing. So our forward primer is located at the 5' of the DNA given. So if we were looking at a sequence, it would be located the 5' end of that sequence, and it's the same as the DNA in the given direction. So if our sequence was really fast, A C C T G A C C G, and we had this was 5', this was 3', and this was our site for the primer. Our forward primer would literally just be 5' A C C T G. So it would be the same copy. It would be exactly the same as this right there. But if we were doing the reverse primer, which we would have over here, the reverse primer is located at the 3' end of the DNA given and is inverse and complementary of the DNA given. So first we would have to flip this. So we actually go for the G first, and that becomes a C, and then G, G, T. So that's it goes something like this. So all of these are translate to one another. And it's also complementary. So we have A goes to T, G goes to C, C goes to G, etc. So that is how you do, that would be the reverse primer, and that would still be 5' to 3', because we, in order to have this 5' to 3', then we can have the DNA synthesized that way instead of synthesizing off that way into part of the DNA that we don't want to copy. So this forward primer tells the DNA to synthesize that way, and the reverse primer tells it to synthesize the other way, because between those two primers is the sequence that we want to copy. And for the products of PCR, we have the original DNA will have 2 copies because it was split and then reproduced, but all of the other copies will just be fragments of exactly what we want between those 2 primers, and we can calculate it using 2 to the n number of times it reproduces. And we can determine if the gene is successfully copied if we run the results of our PCR through a gel. There should be really heavy banding at the exact number of base pairs that our region of interest was. So if our region of interest was at 500, then we would see a really big band at 500 base pairs. Another thing that's really important to biotechnology are plasmids, also called vectors. So basically, we can use these to insert different genes into bacteria, to as one step of cloning, as said above. So this is one tool that we use in the process of cloning. So first, we have to cut the plasmid, usually using a restriction enzyme, which we will get to in a minute. And then we add the gene of interest into the vector with DNA ligase, and that gene of interest was amplified in PCR. So this goes PCR, creating plasmid vector, and then we just keep going down to do the whole method of cloning. But this vector will usually be pre-built, and it will contain an origin, it will contain antibiotic resistance, it will contain sites to insert the gene, and it will contain restriction enzyme sites. So what are restriction enzyme sites? Restriction enzymes are basically enzymes that were found, they were produced by bacteria, and they cut DNA at very specific sequences. Each enzyme has one DNA sequence that it will cut at. And so we can, by using specific restriction enzymes, we can cut the plasmid exactly where we want into fragments, which is called a restriction digest. And by doing that, we can determine if the gene of interest was successfully added or not. So after we check that this gene of interest was successfully added, we can then transfer the vector into the bacteria. And when we do this restriction digest, we will get to checking it later when we talk about gel electrophoresis. But when we do bacterial transformation, we can either use heat shock or electroporation or other methods to basically get the bacteria's membrane to open a little bit so that that plasmid can go in and the bacteria will accept it and start making copies of it and start transcribing it and translating it and expressing the genes. So once we have these in bacteria, we need to check if they have successfully been implanted in the bacteria. And so in order to do that, we can either have a color gene be on the plasmid, which means that all the bacteria with the plasmid will be a certain color and the bacteria without the plasmid won't. Or we could have antibiotic resistance. So when we grow the bacteria on a plate with antibiotics on it, the bacteria that survive, we know have been transformed successfully because they have that gene that was on the plasmid, and the bacteria that weren't transformed successfully are killed. So that's a really good and easy way of making sure that we only have the correct bacteria. So yes, we can also do the restriction enzyme at digest as I talked about before, when we do a restriction digest, we usually have the untransformed vector in a few lanes, and we do the same restriction enzyme digest on it, and it will come out to certain bands. And then we would also do the transform vector, and it might have different bands depending on where the gene of interest was added. If we compare the two bands, we can make sure that everything was added where it should be and that we truly did have the transformed vectors inside the bacteria. So that's just another check we can do. We can also do PCR of the bacterial genome with certain primers to make sure that we have the gene of interest in the bacteria as well. We then have blotting and hybridization, which are different ways of analyzing levels of certain molecules in an organism. So we have the northern blot, which is RNA levels. A good way to remember this is that we have the northern starts with N and it also has R in it, and I think that kind of correlates to RNA. We have the western blot, which is protein levels. Um, I kind of think of that as like Wild West. They like cattle ranchers like their protein, so like Western blot equals protein levels. Again, all of these memorization tricks are a bit of a stretch, but it's whatever you can do to make sure that you know which blot corresponds to what. Southern blot, I think of genomic DNA because in South you might want to do some more genome studies because you never know who you might be related to. So that's a funny way of remembering those, and I hope that helps. But that's just how I remember these. Um, we also have in situ hybridization. In situ means within the cell, and we find all of the RNAs or proteins within an organism in a cell by basically freezing the cell, but keeping everything intact, and then washing it with probes that will attach to any of the desired RNA or protein. And those probes will be labeled with like a radioactive marker. So then when you look at them under a microscope, you can see um, all of the target RNAs or proteins that you're looking for. We also have different libraries, and we have the genomic library, and that's usually the entire genome of the organism. If we are in a cDNA library, that means we're only using coding DNA, and that does not include introns or promoters. And this is basically the DNA transcribed from mature RNA. So everything that was spliced out doesn't get included. We also, as a side note, have hairpin RNAi, which is basically the synthetic version of miRNA and siRNA, which are used to go around and regulate levels of mRNA and transcription and translation. That is here because it was here in the slides, but it has nothing to do with libraries. We also have sequencing, and this is a way we can determine the coding regions of genes, and we can also determine the targets of MI and siRNAs, which I just mentioned about these hairpin RNAis are kind of the synthetic version of these. And we can also analyze promoter regions. We can basically analyze whatever we want out of whatever DNA we want. There are many ways to sequence DNA, but there are old ways of sequencing which could only do about a couple hundred base pairs at a time, and then there's next gen sequencing which can do a lot more and is what most biotechnology companies are currently using. Then we have the idea of transgenic animals, which is basically when we have genes from one organism that is put into a completely other or different organism that is a different species. So, for example, sometimes we put human genes in mice to see how the mice respond to that and how that affects their phenotypes. That would be, again, something of a reverse genetics experiment. But often we do that so that we can test things like drugs and medicines that will be going out that we can't exactly test on humans, but we really need to get concrete data before we can distribute it to the general population. We also have knockout animals, which is when we silence a gene in those animals and see how that affects the phenotype of the animal, which is basically another way of reverse genetics, but just a different way of it. In general, biotechnology is using techniques to create new genotypes or using one species to improve another. So it's a lot of, for example, just what I talked about the mice, it's transferring one gene from an organism to another in order to benefit. In this case, it would be humans. We also can touch on CRISPR, which is a relatively new technology that uses the Cas9 protein complex with a guide RNA to cut the DNA at sites specified by the researcher. And we can use it to DNA, and we can also use it to insert DNA at said cut. So it's a very powerful gene editing tool that's been a buzzword in like recent years. Okay, so now we're going to talk about genomics and proteomics, which are just fancy ways of saying a map of all the DNA in an organism and the map of all the proteins created by the DNA in that organism. So we're really going to be talking about mapping. So genomics is the study of a gene, and basically when we map it, we're trying to locate genes on their chromosomes and create genetic maps with how far apart are all of these genes. And so we have two types of maps. We have genetic maps and physical maps. And so for genetic maps, we use markers such as short tandem repeats. We use linkage studies just like we did in last unit, so calculating the distance between two genes. And we also use banding patterns on karyotypes of a chromosome so that we can tell which genes are really heavily expressed and which are not, because how condensed the chromosome is correlates to how much that gene is expressed in that given time. How much that gene is expressed in that given time frame. We also have such thing as RFLP markers, and that's basically when genome is cut with specific restriction enzymes. Each genome has a different banding pattern, and these banding patterns are actually associated with different alleles, and so we can actually then use this as a kind of DNA fingerprint, and sometimes this is used, a classic example of how this is used is to determine whether people are related, specifically like paternity tests. We can see that these parents are heterozygous for a gene, and then we can see exactly how those alleles are passed down and how those alleles affect the RFLP markers when we do this gel with the children. So when we're looking at this, we see which ones stay the same and which ones are different? And is there a way that we can get a combination of these to produce one of these three options? And so we can often see that if we're trying to compare parentage, and the bars aren't lining up, then that usually means that they're not related, because if there's no way to get to that bar, then it must be a different allele than the one the parent has. Let me draw you out an example. Okay, so this is an example of something that would not make sense. So if you see in this little drawn out gel that we have the two parents and we have the child, and basically this band does not match up with either of the parents' bands. So although this one does and this one does, so we could possibly argue that this person is not related to these two people because it has different banding pattern than any of these two people could provide. The whole thing with RFLPs is that we can compare different alleles without actually sequencing the gene itself. We can compare the alleles between different people and see they have the same alleles or they have different alleles. For these RFLPs, we use short tandem repeats, which is repetitive non-coding DNA, and each person has a certain number of these repeats of specific sequences, but each number is a different allele in the population. So if we compare that this person has this many repeats and this person has this many repeats, they'll actually turn out to have different banding patterns. And then we can use that to compare alleles. We also have the concept of a physical map, which is based on actual distances in base pairs. And this is a lot more precise than a genetic map, and it's basically like new and improved edition. And often uses it uses more accurate sequencing, and we fragment large chunks of DNA. We sequence those chunks of DNA, and then we stitch them back together by seeing which parts overlap with each other. Um, and that we can also do something called shotgun sequencing, which is a little bit faster, and it basically skips the fragmenting part. Um, and we just get the entire genome, toss it in there, fragment it once instead of fragmenting the genome and then fragmenting those fragments and then just sequencing. Sequencing is super useful because we can identify the small variations in DNA from organism to organism, because really, even though our everybody is really wildly different phenotypically, only a very small percentage of our DNA differs from person to person. So we can use this for DNA fingerprinting, and we can also use this for haplotyping. So that's basically looking at how related people in a population are. So we can see the genetic variation based on country. Sometimes certain populations are more susceptible to certain diseases, and we can see what is the genetic variation that makes this population more susceptible to this disease than this population. And a lot of people do that based on countries, based on continents, based on location, really isolated populations versus less isolated populations. So haplotyping is a really interesting study that we can see to compare just the relatedness of people in a certain population and how that population responds to different stimuli, such as an infection or a virus. We also have metagenomics, and that's a way to identify species in a community. So that's like if there's a ton of bacteria and a ton of different bacteria on certain place, we can use it to identify the species of bacteria because there are millions of them, and we honestly don't know a lot of them. So, metagenomics is a good way to, in a big community of organisms, determine, oh, this bacteria is this bacteria. Or even if we have a bunch of different kinds of moss grouped together, we can determine which kinds of moss are correspond to which types of moss. We also have synthetic biology, which is the idea that we can manufacture a custom genome, or a gene to do the function that we want, and that is still a developing field that we're trying to um, go for. But as you might have heard with the kind of dire wolves, this is something that would be kind of in the field of synthetic biology. We also have the functional genomics, which we have basically different types of genomic libraries. So we have the transcriptome, which is the library of all coding mRNAs, and we have the proteome, which is the library of all expressed proteins. And so basically we can look at there and see, oh, we have the genome, and then we can match that with the proteome and see what DNA actually codes for which proteins and what they do. We also have the tool called BLAST, which identifies genes based on their similarity to genes across different organisms. So this compares uh, genes on the level of different organisms, and we can often find homologous genes, which are genes that are similar between different organisms and evolved from a common ancestor. We can also find um, organisms that have the exact same gene with the exact same function. And we can also find genes within the same organism that are basically the same, but they all have slightly different functions. All genes are homologous because all of them have to come from somewhere. If you look at phylogenetic trees, we can see that our homologous genes would be the genes that came from the organisms that these organisms most recently evolved from. We also have genome level studies. So we have microarrays, which are basically we put a ton of single genes or DNA fragments onto a microscopic scale onto a grid, and then we basically wash them with probes to see to mark our targeted gene or to mark our targeted DNA, and we can look at the expression, we can compare it to another microarray that was washed with a different marker, and we can use that to look at stuff like alternative splicing, mRNA, and links to diseases. We also have the idea of comparative genomics. So that's the evolutionary studies of organisms. Um, so that's kind of trying to see, oh, does a bigger genome mean a more complex organism? Which actually, the answer could sometimes be no, because a lot of that could be a lot of non-coding DNA or vestigial DNA. We can also look at the different functions of genes across organisms. And again, going back to like homologous genes, we can trace back where genes originated from and how they've evolved since then. And then there's also the idea of proteomics, which is basically finding all the proteins present in a particular cell, studying these proteins, and we can actually do a 2D gel electrophoresis to look at those proteins and study those proteins. So yeah. All right, and now we are going to be talking about epigenetics. So epigenetics are heritable changes in the genome but do not change the sequence of the DNA. So basically, the sequence of the DNA remains completely unchanged. ACG is still ACG, but there are markers on that DNA or stuff like histone proteins that are heritable that will change the expression of the DNA and therefore the phenotype. So we have many methods of this, and one is DNA methylation. So we've talked about this before, but basically methylating the gene is associated with repression. Demethylation is associated with activation of transcription. And basically when methylation occurs, it can leave marks that stay even after replication and cell division, meaning that it can be passed to actually future generations of this organism. The same thing can happen with histone modification, which happens in a multiple of ways, phosphorylation, methylation, etc. And all of these marks are known as the histone code. And this can also be passed down and is heritable. So, environmental changes that cause this methylation or demethylation or acetylation can actually affect this histone code and be passed down. So the environment which your parents lived in actually affects how you are today. Um, RNA can also affect histone code because it can regulate transcription initiation. We also have paramutation, which is when we transfer an epigenetic state or marker, so like a methylation marker, to another gene on a different locus. So if you had, say, a marker on chromosome 15, if it suddenly the marker was transferred to chromosome onto a locus on chromosome 16, then that would be something that would be an example of paramutation. It doesn't have to be across chromosomes, just across gene locuses. Um, and then we also have behavior. So early childhood environment stressors can actually influence the behavior of when you grow up as an adult. So it can affect stuff like stress levels, it can affect your tendency for having abnormal psychology when you grow up, and it can also impact stuff like chronic illnesses that can come on from having too much stress at an early age. Um, we can also observe this in when we have identical twins and when there are differences between them, whether especially phenotypically, these are likely due to epigenetics. Epigenetics can also affect which X chromosome is inactivated during X inactivation. But a lot of these processes of epigenetics are not super well understood. So this is still a huge field that we're trying to research. We also have genomic imprinting, which means that the sex of one parent of the allele determines whether the allele is expressed. So if we have maternal imprinting, that means only the allele of the mother is expressed. And this often occurs in gene clusters, and the gene of the other parent will be silenced by non-coding RNAs, meaning that the other parent's allele is the only allele expressed. We also have the concept of an epigenome, and that's kind of like a library of epigenetic marks that affect gene expression or the phenotype. So just like we have a genomic library, that's a library of all of the base pairs that are in a DNA, or proteomic library, which is a library of all the proteins that are created. We also have an epigenomic library, which shows all of the different markers um, that affect gene expression, and specifically histone modifications, ones that are specific to development, and marks that actually determine other epigenetic marks. We also have this concept of immunity, and so this is kind of a side tangent to the rest of the unit, but we still covered it. And so basically, your ability to fight off an infection depends on genetic variability, and this genetic variability is generated by the individual throughout their lifetime. So we have these things called B cells, and as you go on with life, these B cells will keep reproducing and recombining and creating a bunch of variations in these antibodies. And these antibodies, when they match up with an infectious cell, then we make a ton of clones of this one antibody so your body can fight it off. And then once that infection is over, we keep some of them in memory, and that's basically how our immune system develops and grows and builds. Um, Immunoglobin is just another word for antibody, and they have a Y shape, and they have two heavy chains and two light chains. And basically, the moral of the story for this class is that they have highly variable regions, and that is due to recombination in these genes when these B cells are being made. And also, this is an example of how mutations can be good, because when you have mutations in these genes, that's how we can create more variability, so we have the potential to fight off more diseases. All right, so this is the end of this review video. Thank you for listening, and good luck studying.